Welcome back to the Deep Dive. Today, we're taking a really high-speed journey, sort of into the engine room of modern ag and biotech. We're moving past, just traditional crossing and getting properly genomic.
Absolutely.
Our mission today is to really understand two revolutionary tools that are driving genetic progress. Genome-Wide Association Study, that's GWAS, and its powerful partner, Genomic Selection, or GS.
Yeah, these are really the technologies defining the speed of modern breeding programs. Doesn't matter if you're talking corn, wheat, dairy cattle.
Right.
They let us make genetic decisions in, well, months instead of the years it used to take.
And the reason this speed up is so critical, it really comes down to a fundamental idea in breeding, doesn't it? The breeder's equation. Conceptually, it tells us that the speed of improvement depends on, well, a few things: how intensely we select, how heritable the trait is, how much variation we actually have to work with.
Right. Selection intensity, heritability, variation.
But crucially, it's inversely related to time, the time it takes to complete one breeding cycle.
Exactly. That's the key variable these methods tackle. If we can shave years off that generation interval, the L in the equation, the overall rate of gain just skyrockets.
Because traditional breeding meant waiting for plants to grow, flower, measure them, reproduce.
All that time. Time we often don't have if we want rapid progress. GWAS and GS, they let us identify the good genetics, sometimes before the seed even sprouts. Cuts out huge chunks of waiting time, makes selection dramatically faster.
Okay. So let's unpack this. Maybe start by laying the groundwork. If you're trying to connect a trait, like disease resistance, to a specific spot on a chromosome, you need variation, obviously. Traditionally, the main approach was linkage analysis, right? Often called QTL mapping. And this uses very controlled families.
Yes, mapping populations. They come from specific controlled crosses, think like an F2 population, or recombinant inbred lines, RILs, derived from just two specific parents usually.
So it's very tidy in a way.
It is. The genetic relationship is pretty straightforward. But because you're typically only looking one or maybe two generations after that initial cross, you actually have relatively few recombination events. Not much natural shuffling has happened yet.
Okay. So the link between your marker and the gene might be strong, but the actual region on the chromosome you flag is still pretty big. Hard to pinpoint the exact gene.
Precisely. Your detection power for QTL is high, but your resolution finding the actual gene is generally poor.
Right. So now contrast that with GWAS, which uses diversity panels. What are those?
So that's where the big shift happens. Instead of that narrow controlled family from two parents, GWAS uses much broader collections. Think germplasm banks, land races, maybe a panel of hundreds or thousands of diverse, often seemingly unrelated individuals.
And the fascinating thing here, as I understand it, is that GWAS doesn't rely on just the few shuffles, the recombinations we created in the lab.
No, not at all. It leverages all the historical recombination events, everything that's happened naturally over potentially hundreds, even thousands of generations of evolution within that diverse population.
Which brings us right into our first section then. Association mapping, or GWS. The search for specific, powerful markers. Why is using all that historical recombination such a big deal?
Because it massively increases the potential resolution. Think about it. Over all those generations, there have been countless chances for a marker that's near the causal gene, but not the gene itself, to get separated by a recombination event.
Okay. So genes that started out close together have had more opportunities to get shuffled apart.
Exactly. This allows us potentially to pinpoint the location of the gene much more accurately than we typically can with linkage mapping in a biparental family.
But this high-res detective work, it hinges entirely on this concept called linkage disequilibrium, LD, right?
Yes, LD. It sounds complex, but it's basically just saying that certain versions of genes or alleles are inherited together more often than you'd expect just by random chance. There's a non-random association.
Like genetic hitchhiking.
That's a great way to put it. A specific marker allele, which might be easy to measure but doesn't actually do anything for the trait itself, is physically close enough on the chromosome to the actual causal gene, the QTL.
So they travel together?
They hitch a ride together through the generations more often than not. It's this statistical covariance, this non-independence that lets us use the easy-to-find marker to track the important but maybe harder-to-find gene.
Okay. So physical closeness is the main driver.
It's the easiest cause to understand, definitely. But we have to remember LD is also shaped by history. Things like selection pressures favoring certain combinations, random genetic drift, mutation rates, even how the population is structured. They all influence the patterns of LD we see.
And how fast that hitchhiking relationship breaks down, that LD decay, that determines how many markers you actually need.
Absolutely critical. If LD decays very quickly, meaning markers and genes separate rapidly over short physical distances, like you might see in really diverse maize landraces, sometimes less than 1,000 base pairs.
Wow, tiny distances.
Yeah. Then you need a huge number of markers, maybe hundreds of thousands or millions, spread across the genome to make sure you have one close enough to catch the association before it breaks down.
But in other crops?
Well, take something like soybean, a self-pollinator. LD often extends over much longer distances, maybe millions of base pairs.
So the hitchhiking block is much bigger.
Right. Which means you need fewer markers overall, because pretty much any marker within that large block is likely to still be associated with the trait gene.
Okay, this is where it gets really interesting for me, though. Because using these diverse panels and relying on historical events sounds powerful for resolution. But it must introduce some big statistical challenges. Spurious associations.
Oh, absolutely. That's the biggest hurdle in GWAS, false positives. And the main culprit is usually population structure.
Okay, what does that mean in practice?
Well, imagine your diversity panel isn't one uniform group. It's actually made up of, say, 2 or more distinct subgroups that haven't mixed much historically, maybe tropical varieties and temperate varieties.
Right.
And let's say, just by chance, or maybe due to past breeding history, the tropical group tends to have higher yield and also happens to have a high frequency of a particular marker, allele, say, allele A.
Right.
Your GWAS analysis will see that allele A and high yield often appear together and flag it as a significant association.
Even if that marker has absolutely nothing genetically to do with yield itself.
Exactly. It's a false positive driven by the shared history or structure of that subgroup, not by true genetic linkage between the marker and a yield gene. The classic confounding problem.
So you have to somehow filter out that background noise, that historical static, to hear the real genetic signal. How do breeders do that?
We rely on some pretty sophisticated statistical tools, specifically linear mixed effect models. The most common framework you'll hear about is the Q plus K model.
Q plus K.
Right. It's specifically designed to try and tease apart those false signals caused by structure and relatedness from the true genetic associations we're looking for.
Can you break down Q and K for us? What do they actually represent? Conceptually.
Sure. Think of it this way. Q basically represents that large-scale population structure. It tries to account for those distinct subgroups, our tropical versus temperate example, or sometimes visualized as different clusters of dots on a graph.
Okay.
We usually treat this as a fixed effect in the model. We're trying to specifically estimate and remove the average effect associated with belonging to one group versus another. We often infer this structure using methods like principal components analysis, PCA, based on the marker data itself.
Okay, so Q handles the big group differences. What about K?
K stands for kinship or relatedness. Even within those broad subgroups identified by Q, individuals are still related to each other to varying degrees. Some are like cousins, some are more distant. This reflects more recent shared ancestry.
Right, it's not just about the ancient splits.
Exactly. And we model this kinship as a random effect. The K-matrix, often called the relationship matrix, quantifies how related every individual is to every other individual in the panel, again, usually estimated from the genomic markers.
So by fitting both Q structure and K, kinship, in the same model.
We essentially try to account for the baseline similarity between individuals due to both deep history Q and more recent family ties K. By doing that, we can better isolate the additional effect of a specific marker that might be genuinely linked to the trait, over and above all that background relatedness. It helps clean up the results, giving us a more reliable Manhattan plot with fewer false peaks.
That's crucial. It moves us from potentially seeing associations everywhere to hopefully just seeing the true hits. Okay, so GWAS is like this high-powered microscope, great for finding specific genes, often ones with pretty big individual effects.
Generally, yes, it excels at discovery of major QTLs.
But let's shift gears now to genomic selection, or GS, because this feels like a different beast altogether. It's less about discovery and more about prediction on a massive scale.
That's a perfect way to put it. That conceptual shift is really vital. While GWAS is fantastic for finding those major genes, most of the traits we really care about, economically think, Overall yield, stress tolerance, feed efficiency, nutritional quality, they're highly quantitative.
Meaning lots and lots of genes are involved.
Hundreds, maybe thousands, each with a potentially tiny effect. They all add up to the final phenotype. GWAS might find the few big players, but it misses the combined effect of all those small effect QTLs.
So if you only select based on the few big genes GWAS finds, you're leaving a lot of genetic potential on the table.
A huge amount. So genomic selection basically says, look, let's stop trying to find every single needle in the haystack.
Forget identifying the individual genes.
For the purpose of selection, yes.
Yeah.
Instead, let's use a really dense set of markers covering the entire genome. Thousands, tens of thousands, hundreds of thousands. And the goal isn't to pinpoint which marker is next to which gene. The goal is to use all the markers together to capture the cumulative effect of all the QTLs, large and small, influencing the trait across the entire genome. We want to predict the total genetic merit of an individual.
Wow. Okay. But using that many markers, surely that creates its own statistical headache. I remember hearing about the big P, small n paradigm. Mewison et al., 2001.
Yes, the classic paper. That's the core statistical challenge of GS. P is the number of parameters you're trying to estimate in this case, an effect for potentially every single marker. N is the number of individuals you have actual phenotype data for.
And in GS, P is often way bigger than N. Lots of markers, fewer measured individuals.
Exactly. You might have 50,000 markers, P, 50,000, but only, say, 2,000 plants or animals with reliable yield data. N equals 2,000.
What happens if you just try and analyze that with standard statistics like ordinary least squares, OLS?
Complete disaster. OLS tries to find the single best effect for each marker independently. With far more markers than observations, it essentially finds spurious associations and fits the random noise in your specific data set perfectly.
It overfits.
Massively. The model looks great on the data used to build it, but it's completely useless for predicting the performance of new individuals. Its predictive ability is 0 or even negative.
So OLS is out. What's the solution?
The solution is to move away from treating marker effects as fixed unknowns that we estimate independently. Instead, we use methods involving penalized or shrinkage estimation, typically within a mixed model framework. We treat the marker effects as random.
Random effects, how does that help?
By treating them as random variables drawn from some distribution, usually assumed to be centered around 0, we essentially force the model to be more conservative. It shrinks the estimated effects towards 0, especially for markers with weak evidence.
So it prevents any single marker from getting too much credit just by chance.
Exactly. It forces the model to borrow information across all the markers, leading to more stable and reliable predictions. It smooths things out and reduces overfitting.
And this leads to the actual models used in practice, like ridge regression, BLUP.
RBLUP.
Yes, RBLUP is one of the most common starting points. It assumes all marker effects are random and come from the same normal distribution with a mean of 0 and some variance. The ridge regression part refers to the type of penalty or shrinkage applied. The BLUP stands for Best Linear Unbiased Prediction, which is the statistical method used in mixed models to predict these random effects.
And it's simpler because
Because you only need to estimate one single variance component, the variance of that distribution from which all marker effects are drawn. The single parameter controls the amount of shrinkage applied uniformly across all markers. It's computationally pretty efficient.
Okay. And then there's its close cousin, GBLUP, Genomic, BLUP. Right. GBLUP is conceptually very similar, but mathematically slightly different, and often even more efficient computationally. Instead of explicitly modeling the effect of every single marker, GBLUP models the total random genetic effect, let's call it U, for each individual.
Okay, the overall genetic merit.
Yes. And the key insight, particularly from Van Ryden's work in 2008, was that the relationships, the covariances between these U values for different individuals can be estimated directly from the marker data using the genomic relationship matrix, which we often call K.
Ah, the K matrix again, the same one used potentially in GWAS.
Well, conceptually related, yes. It quantifies the pairwise relatedness between all individuals, but based on their actual shared markers across the whole genome.
And this is powerful because it reflects the realized genetic relationship, what they actually inherited.
Precisely. Not just the expected relationship based on a pedigree chart, who the parents and grandparents were, which we call the A matrix. The K matrix captures the actual outcome of meiosis, the random shuffling, any errors in the pedigree. It's the ground truth of relatedness based on the genome itself.
That seems incredibly important, using the real relationships.
It is. And this GBLUP approach, using the K-matrix, forms the core of how GS prediction often works in practice. It requires setting up a specific workflow, usually involving 3 key populations.
Go, what are those?
First, you need the training population. This is your reference set. These are the individuals for whom you have both genotype data, the markers, and phenotype data, the actual measurements like yield, height, disease score, etc. This is the expensive part, getting the phenotypes.
And you use this group to build the prediction model.
Exactly. You train the GBLUP model, essentially figuring out the link between marker patterns and performance in this group. Second, you often have a testing population or validation set.
To check if the model works.
Right. You use individuals with known phenotypes but pretend you don't know them. You predict their genetic merit, their genomic estimated breeding value, or GEBV, using the model trained on the training set, and then you see how well those predictions correlate with their actual measured performance. This gives you an estimate of the model's predictive ability or accuracy. Cross-validation techniques are key here.
Makes sense. Train, then test. And the third population?
That's the selection population. These are the actual candidates you want to make decisions on, often young seedlings or animals early in life. For these, you only need their genotype data.
No phenotyping needed for them.
Not necessarily at this stage. You just get their marker data, run it through the prediction model you built and validated, calculate their GEBVs, and then you select the individuals with the best predicted genetic merit to become parents for the next generation.
And because you skip the lengthy phenotyping step for this selection group, you can make decisions much faster.
Dramatically faster. You compress the breeding cycle, sometimes selecting purely based on DNA as soon as you can get it. That's the big win for accelerating genetic gain by reducing the generation interval.
But that accuracy, the predictive ability you mentioned, that must depend heavily on a couple of things.
Absolutely. It hinges on the level of LD between your markers and the actual QTLs you need, markers that are reliably tracking the genes. But maybe even more critically, it depends on the relationship between the training population and the selection population.
They need to be related somehow.
They need to share a similar enough genetic background and LD patterns. If your training set is genetically very different from the group you're trying to predict, well, the predictions won't be very accurate. It's like trying to use a Spanish to English dictionary to translate a German document. The relationships learned in the training set have to be relevant to the selection candidates. The composition of that training set is vital.
Okay, so let's try and bring this all together. We've got GWS and QTL mapping.
Essential for discovery. They're the tools to find specific genes, especially those big hitters with large effects that we might want to target directly, maybe for marker-assisted selection or even gene editing down the line. The microscope, as you said.
Right. And then we have genomic selection,
which is the large-scale predictive engine. It doesn't worry so much about identifying individual genes, but aims to capture the cumulative effect of everything, all those tiny QTLs included. Its power comes from accurately predicting overall performance and enabling much faster selection cycles, dramatically cutting down that generation interval. It's about accelerating gain for complex traits.
And in practice, even though there are more complex models out there, like Bayesian methods?
Yes, things like Bayes-B, Bayes-C, lots of variations exist.
The simpler ones, GBLUP and RRBLUP, are often the workhorses.
Very much so. For many situations, especially in plant breeding, they provide predictive accuracies that are very similar to the more complex methods, but they are computationally much faster and easier to implement. So they tend to be the backbone of many large-scale breeding programs. They deliver the goods efficiently.
Okay. That provides a really clear picture of these two powerful genomic tools. Which brings us to our final thought for you, the listener, to maybe ponder. It relates back to that crucial genomic relationship matrix, the K matrix. We've established that K gives us the realized genetic relationship based on the actual markers shared between individuals. And this can be different from the expected relationship based on pedigree records, the A matrix.
Right. Pedigrees assume certain inheritance patterns, and they can have errors. K reflects reality.
So here's the thought. If you're running GS in a valuable, established breeding population, perhaps one with decades of pedigree records, and your analysis consistently shows a significant widespread difference between the realized genomic relationships, K, and the expected pedigree relationships, A, what does that really imply? Not just about the underlying genetics, but perhaps about the history, the integrity, or maybe just the inherent complexities and assumptions within the record-keeping and management of that population over time.
It's a fascinating question about what the genome reveals versus what the records say.
Something to think about. We'll leave that with you.